Hey everyone. So in this lecture, we're going to look at what is likely an inefficiency in our plants, and it's this process of photorespiration. And we're going to go through the process. We're going to compare it to normal photosynthesis, and then we're going to look at some methods or a method that our plants have developed to counteract this process. So before we get into photorespiration, we are going to review the Calvin cycle of photosynthesis. And if you remember the Calvin cycle, we also called it the dark reaction or the light independent reaction. And it is that process where our plants are taking carbon dioxide, and from that carbon dioxide, they are making sugar or glucose. Right? And if you recall, the first step in this Calvin cycle was that our carbon dioxide combined with a molecule called rubisco bisphosphate, or RUB-BP. Right, and in actuality, it's 6 carbon dioxides and 6 of these RUB-BPs. And if you remember, when we were dealing with the Calvin cycle, we like to keep track of our carbons. That's like a nice, easy way to keep track. Right, so this RUB-BP is a 5 carbon molecule over here. This is our 1. And then I gave you kind of a simplified version where from here we had the development of 12 PGALs, and these are phosphoglyceraldehyde. If I spelled that right. And then and these are 3 carbon molecules, right? So if we're keeping track of our carbons here, we have 6 times 5 is 30, plus another 6, that gives us 36. And that makes 12 3 carbons. So 12 times 3 is 36, so our carbons are all accounted for. And then here, we had 2 of these guys branch off, and they made glucose, or 1 glucose molecule, so that's C6H12O6, right? And that's the kind of the goal of this process, to make that sugar molecule. And then the remaining 10 went back and they formed these 6 RUB-BPs again, right? And it's a cycle, so this can be repeated indefinitely. So I mentioned here, and also this right here, just so we're being accurate, this is going to take ATP and that NADPH, which are produced in the light reaction from that electron transport chain. I think it takes 12 of each, right? And then they produce ADP afterwards, and NADP plus. Right, so it takes this, really takes energy for this process to make these PGALs, and then it makes our sugar molecule, and we can repeat this process. So this was a simplified Calvin cycle, because there are actually several intermediary steps to get to this PGAL. And we're not going to go through all of them, but I do want to mention one because it's fairly important when we talk about photorespiration. So technically, the first thing that is created is something called we can call it 3PGA, or 3-phosphoglycerate. Again, my handwriting is awful, but that's technically the first compound that is created, or molecule that's created, 3-phosphoglycerate. And this guy is a 3 carbon one as well. And then there's a couple other intermediate steps, but eventually get to this PGAL. So when we went through this originally, I kind of skipped these beginning ones. But I want to bring it up now because it's going to play a role, as we'll see, in photorespiration, or at least help us in understanding photorespiration. And this is sometimes referred to as C3 photosynthesis because the first molecule that is created has 3 carbons, right? C3. Now, I also didn't bring up the enzyme that kind of allows this whole process to occur. And that enzyme is called Rubisco, right? And the actual full name of this, just so you know, is ribulose bisphosphate carboxylase oxygenase. Right? Really big name. It's why everyone just refers to it as Rubisco. So this is the enzyme that is catalyzing this entire reaction, right? It allows this reaction to occur. It is taking this RuBP and combining it initially with this carbon dioxide to try to kick off this cycle. Now, an issue with this Rubisco is that, and you can kind of get it from the name, right? Carboxylase makes you think carbon dioxide is being combined with this ribulose biphosphate, but it also has oxygenase in the name. And essentially what happens in photorespiration, and I'm going to do it over here, is that this Rubisco can not only fix carbon dioxide, and by fixing, remember, it means we're taking it from being a gas and making it into an organic compound, right there, our glucose molecule. So it can not only fix carbon dioxide, but it can also fix oxygen. And when it does this, it's actually this inefficient process where we wind up losing carbons, right? And we don't produce any glucose. So let's take a look at that process now. Okay, so, and really this can occur when oxygen is present in our leaf, right? And that can occur depending, there are different conditions that can create more oxygen. But, you know, while our plants are conducting photosynthesis, occasionally there's going to be oxygen there, and this Rubisco can fix that oxygen instead of carbon dioxide. So what happens here with photorespiration is we have oxygen that is starting this process, right? So instead of our carbon dioxide, we have our 6O2. And just like with regular Calvin cycle, a first step is that it combines with our 6-RuBP, right? And here's where we get something different. So instead of creating first 12 of these 3 PGALs, I should do that, or 3 PGA, so instead of creating 12 of these phosphoglycerates, what happens is, in this process, we only get 6 of these phosphoglycerates, 3 PGA, and it also produces, which again, these are 3 carbons, it also produces 6 phosphoglycolates. And I don't know if there is a abbreviation for that, so I'm just going to write it out. Right, and this is a 2 carbon. Right, so automatically we have something different here in this process. Right, so instead of our RuBP combining with carbon dioxide, it's combining with oxygen. And as a result, we're only producing 6 of these 3 PGAs instead of 12. And then we're producing 6 of this other molecule called phosphoglycolate. And if we're keeping track of our carbons like we should, again, here's our 5 carbon guy over here. So instead of having a total of 36 carbons to start, now we only have 30. Right? And we go through this here, and we have now 6 times 3, and then 6 times 2. So 6 times 3 is 18, 6 times 2 is 12, matches up, we have our 30 carbons. Now, again, the process, what our plants ultimately want to do with photosynthesis is to create glucose, and they need, really you need 12 of these PGALs to do that. But now we only have 6 of these, right? So we're missing. Now, our plants have evolved what people refer to as a salvage pathway, and it's this complex pathway where it takes these phosphoglycolates and it's able to convert some of them back to these 3-PGAs, these phosphoglycerates, right? So it goes through this whole complex process. We're not going to go into it, but ultimately we're able to get 3 more of these back, which is something. And in this process, we also produce 3 carbon dioxide, right? So now we have a total of 9 of these PGAs that will result in 9 PGALs. But here we're at a deficit, right? We are at a deficit of 3. So we have no PGLs that go to make glucose. Everything goes back to try and make as much Rub BP as we can, but again, we can't complete this cycle because we need 10 of these if we want to actually make what we lost. So as a whole, we are losing carbons and we're losing energy because this is taking ATP, right, to power all this stuff. So we're losing energy. We're not producing any sugars, right? And we're losing carbons in this process. So this could not be repeated indefinitely. So it's a big inefficiency, right? It's not, if we are trying to maximize the amount of sugars that we're creating as a plant and minimize our energy costs, this is not the way to do it, right? And this is, I'm going to write up here just so it's clear, this is photorespiration. So given the inefficiency of this process, people wonder, why is this even around, right? Why did this evolve? Why is it still present in our plants? And there are a number of theories, right? So photorespiration might be this legacy of the evolution of photosynthesis way in the past. Right? So when our plants and algae kind of start evolving and they start undergoing photosynthesis, at that time, the amount of oxygen in the atmosphere was very low, right? So and I should say this is all, I should write all like here, this is that Rubisco. Right, that's the enzyme that is, carrying out this entire process. But anyway, at the time that this evolved, it was likely there was very little oxygen in the atmosphere, so this process wasn't a big deal, right? There wasn't a lot of, I'm going to say Y here, right? So there was low O2 when plants evolved. Right, so there was low O2, there wasn't a lot of oxygen around, there wasn't a lot of, there wouldn't be a lot of instances of this photorespiration, so it wasn't a big deal. There wasn't a lot of selective pressure on our plants to get rid of this process. Now, obviously now that has changed, right, and there is we do have more oxygen in the atmosphere, partially due to, or actually completely due to our plants photosynthesizing. So why does it exist now? Well, it may just be a holdover of that process, right? And it's an inefficiency, but it's not so inefficient that it really causes our plants to die out before they can reproduce. And there may be some benefits to this process. It may be a way of ridding the leaves of excess oxygen. When there's a lot of oxygen in the cellular environment, it can interact with ATP and it can cause damage to the cells. So as you see in this process, it does get rid of oxygen and converts it through this long pathway to carbon dioxide. So this could be a way of getting rid of excess oxygen that could otherwise damage cellular tissue, right? So there may be some benefits. Likely, there's probably some truth in both of these, right? This is a legacy, a holdover from when this process first evolved, and there is enough of a benefit to it that it persists in our plants. So that is photorespiration. And in what we're going to look at now, I was going to say in the next video, but I think I'll combine them all into one. What we're going to look at now is a process by which some of our plants have adapted to deal with this process. Right? And that is through a process of photosynthesis that we call C-4. So C-4, photosynthesis, right? Or you'll hear plants called as, it's supposed to be C4 plants, right? And this is a process it's a process to deal with photorespiration. So it's going to be, I'll write it out there, how plants deal with photorespiration. Right, and as we just learned, the problem with regular C3 photosynthesis, right, I'll pull it back up here. The problem with this process is that this Rubisco enzyme, when oxygen is present, will occasionally fix oxygen as opposed to carbon dioxide. Right, and when it fixes oxygen, it's this really inefficient process that we just looked at. So that is what our plants need to address, right? And ideally, they can do that if there was just an oxygen-free environment. Maybe like when this first evolved way back eons ago, when there wasn't a lot of oxygen around and it was just carbon dioxide. But our plants can't do that, right? They live on the earth where there is oxygen present. So how do they deal with photorespiration / the problem of oxygen? So they've actually come up with this very clever process. And I say come up, this is evolved, but, and we're going to look at it now. So it starts the same, right? And actually, before I get into the process, let's take a look at our leaf anatomy, if you guys remember, right? We learned this way back in the beginning. And if we do like a cross section of our leaf, this is the top of the leaf and the cuticle. And we'll have little openings there, right? Yeah, so up here, this was the cuticle. That is cuticle, right? That waxy covering of our leaf. And then we had some veins that run through there, and that's the xylem and phloem tissue. And then we had tissue kind of like cells like this, right? And this is mesophyll tissue, and it's palisades mesophyll and spongy mesophyll, but mesophyll. So these cells, the mesophyll cells, is it one L or two? I think it's two L's. And in these mesophyll cells, this is where photosynthesis is taking place. At least that's C3 photosynthesis. Right? This is where, in the mesophyll, we'll say, site of photosynthesis. Right? And that is referring to the cells. We know that photosynthesis is taking place technically in our chloroplasts, in those organelles. Right? But in our mesophyll cells is where photosynthesis takes place. Now there are also, and these openings down here, if you recall, these are the stomata. Right? So those are the little openings typically on the bottom of the leaf that allow gases in and out. So as a result, here, all these mesophyll cells, in between them, this is all oxygen and carbon dioxide. So it's both gases are present within here that are able to come in and out of these cells. So because of this, these cells are exposed not only to our carbon dioxide, but also to our oxygen, which we said is a problem sometimes with our photosynthesis. Now, there are cells further in the leaf that kind of ring our vascular tissue, our xylem and phloem, and those are called bundle sheath cells. Now these bundle sheath cells are deeper in the leaf, and as a result, they aren't exposed to gases. They're not exposed to carbon dioxide or oxygen. And that is going to play a role in our C-4 photosynthesis, as we're going to see. All right, so we got a little anatomy background here, and then let's take a look at how this all works. So again, the light reaction is all the same, and we're going to focus on our light independent reaction here, or our Calvin cycle. In this case, it's not quite the Calvin cycle. But the inputs remain the same, right? We start with our 6 CO2. And what is the big change that we're going to see is that instead of that Rubisco, we have a different enzyme called PEP. Carboxylase. Right? So it is not that Rubisco. And what is first being combined with our CO2 is PEP. And PEP stands for phosphenyl pyruvate, right? If you can read that. Phosphenyl pyruvate, that is this guy right here, PEP. Now, again, we can just refer to it as PEP, right? You don't need to know phosphenyl pyruvate. So, what's happening here, and actually to make this easier, we're not even going to worry about the number. Right? We're just going to go through the process. So you don't really need to know about the number of carbon dioxides and PEPs for this one. Right, so this first stage is our PEP is going to combine with our carbon dioxide. Now our PEP is a 3-carbon. We will keep track of our carbons a little bit. Our PEP is a 3-carbon. Our carbon dioxide, again, has 1 carbon. And the first thing that is produced in this process is a 4-carbon molecule called oxaloacetic acid, or oxaloacetate. Right, that's the first product of this, and that is a 4-carbon one. Right, and then this oxaloacetic acid, or oxaloacetate, is going to turn into, or gets it's turned into malate. And again, malate is a 4-carbon molecule. And then from malate, we can produce carbon dioxide and more PEP. Right, so this is the C-4 cycle right here. And you may be wondering, what is the purpose of this, right? Because we started with carbon dioxide, and at the end of this entire cycle, we end with carbon dioxide, right? So we haven't really changed much here. However, when you think about where this is taking place, it's going to start making sense, right? So the end result of this is we can produce carbon dioxide and more PEP, but we're really focused on this malate right here, right? So what is occurring is in our mesophyll cells, which are exposed to the atmosphere, right, or at least some of the atmosphere, so they're exposed to CO2 and O2. So in our mesophyll cells, this C4 is taking place. And the end result, or at least part of the end result of this C4, is this malate. And this malate then gets shipped off over to these bundle sheath cells. And within these bundle sheath cells, remember that within there, in those bundle sheath cells, they're not exposed to the atmosphere. so they're not exposed to carbon dioxide or oxygen, and that's important. So this malate gets shipped off to those bundle sheath cells, and as a little aside, it moves through the plasmodesmata. Remember, if you remember from way back at the start of this course, plasmodesmata are those little channels between our cells, right, that link them together. So through all these channels of these linking cells, this malate gets shipped. Malate? Yeah, you can't see that, but malate goes from cell to cell, eventually making its way to these bundle sheath cells. And within our bundle sheath cells, there is no carbon dioxide or oxygen. But luckily, our malate can break down into carbon dioxide. Right? So our malate builds up in our bundle sheath cells, and there it transfers into carbon dioxide and back into PEP, and that PEP goes back out to our mesophyll cells. But here, we now have an environment within our bundle sheath cells that only has carbon dioxide. And now we can do normal C3 Calvin cycle photosynthesis here without having to worry about oxygen, right? And that's the whole purpose of this thing. We need to create an oxygen-free environment. And we can do that by using this malate. So we go through this C-4 process, we produce malate. That malate then gets shipped off to cells that aren't exposed to the atmosphere, so they're not exposed to carbon dioxide or oxygen. There, that malate can kind of change and release carbon dioxide as one of its products. And now we have an environment that is entirely carbon dioxide, and we can do regular C3 photosynthesis without having to worry about photorespiration at all. It's actually a really neat process and a really neat way of dealing with this photorespiration problem. So some plants that do this are corn and sugarcane, and you think about both, they have high sugar content, right? Because they are very efficient with their glucose production, right? They are producing glucose in these bundle sheath cells. That's where they're doing their photorespiration, and all of their photosynthesis, and all their photosynthesis, they are just converting carbon dioxide. They're just fixing carbon dioxide. They're not doing any photorespiration. So, two C4 plants. Keep writing a G. Right, that take advantage of this adaptation. Right, so that is C4 photosynthesis, and it is a way where our plants are able to deal with that process of photorespiration. Now, the last thing that we are going to look at are CAM plants. Now, our CAM plants, they are essentially, this is a type of specialized C4 process. So CAM stands for crassulacean acid metabolism. Right? Crassulacean acid metabolism. Now, our CAM plants are desert plants, typically, right? And what are desert plants. So if you think back to, again, let's talk about our leaf. So here's our leaf, and we know at the bottom, typically at the bottom of our leaf, are those stomata, right? And these are the pores that open up that allow gases to come in and out, right? So it lets CO2 and oxygen in. But when the stomata are open, it also allows for transpiration, or the evaporation of water. Now, if we are a desert plant, right, we want to try and conserve our water. And one way to conserve our water is to only open those stomata when it is cool out. So opening those stomata during night, right? If you're opening new stomata in the heat of the day, you're going to have a lot of transpiration. You can lose a lot of water. The problem with this is that photosynthesis only occurs during the day. So let's write some of these things out, right? So our cam plants, they want to keep stomata closed during the day to reduce transpiration slash water loss, but photosynthesis only occurs during the day. And for photosynthesis to occur, you're going to need CO2 for photosynthesis. Right, so this is the issue our plants are facing, right? Our desert plants in particular, right? Photosynthesis only occurs during the day, and that's a process where they're creating their food, right? So this is only happening during the day. It's why the dark reaction is a bad name, because even though it's called the dark reaction, it happens during the day. It just doesn't require light. So photosynthesis is only going to occur in the day. In order for that to occur, you need carbon dioxide. In order for carbon dioxide to get into your leaf, you need to have open stomata. But if you have open stomata during the day, you are going to lose water, and you're going to lose water, and potentially it could be a big detriment, especially if you're in an area like a desert that doesn't have a lot of water, right? So you want to try and conserve your water. So it's almost like a catch-22. What are our desert plants supposed to do? Well, luckily, they have developed this process that we refer to as CAM, or crasulian acid metabolism. So what's happening here is our plants are doing that C-4 photosynthesis, but they are doing it during different times, right? So during the night, so at night, they keep their stomata open, right? Now they're keeping their stomata open. It's nighttime. They don't have to worry about water loss so much, right? So at night they keep their stomata open, and they perform. So at night, that's C-4 photosynthesis. Right, so we have, and this is occurring in our mesophyll cells that we looked at previously, and they're performing the C-4 photosynthesis, so they're producing that malate, right? So C-4 photosynthesis, and that is producing malate. Now they store this malate in vacuoles in the cells, and then during the day, actually at night here we should say open stomata. During the day, they close their stomata, and then they take that malate and they convert it back to CO2 and PEP, and then they use that CO2 and they do the normal Calvin cycle, right? So they are, in this way, by kind of breaking up this process, by utilizing the C4 photosynthesis, they're able to deal with this issue where they want to keep their stomata closed during the day, but they need access to carbon dioxide. So they're able to produce carbon dioxide, or at least a molecule that is going to turn into carbon dioxide, during the nighttime when they can keep their stomata open so they can take in this carbon dioxide, they can fix it into that malate, they store that malate, and then during the day they convert that malate back to carbon dioxide and some PEP to kind of go through this entire cycle. And then now their stomata are closed, they have to worry less about water loss, and they're able to do, they have carbon dioxide still that they can then do the normal Calvin cycle. So those are our CAM plants, and it's a subset of our C4 plants. And the C4 plants are really a great adaptation to deal with, in the case of regular C4, to deal with the issue of photorespiration and oxygen in the environment. And it's been adapted further to then deal with these issues of water loss for our desert plants. So really, really neat process. So with that, that concludes our lectures on photosynthesis.